Okay. Jerry, Carl, whenever you guys are ready.
All right. Well, thanks everyone for joining us over your lunch hour. My name is Jerry Clarke. I'm the Chippewa County Crops and Soils Educator with the Division of Extension with UW-Madison. And helping today also co-presenting would be Carl Duley. So Carl, I don't know if you want to introduce yourself real quick.
Sure. Carl Duley, Ag Agent in Buffalo County.
So do have a few slides we'll run through. Feel free to maybe Stefan can monitor the chat a little bit, or just feel free to holler something out with a small group like this. That'll be fine. And if I share my screen here, we can get started with find the right thing here.
I don't know about you, Jerry, but I'd rather take questions as we go along if people wouldn't mind.
Yeah, absolutely. Yep. Just holler them out as you need. So everybody seeing that okay? Come
Yep.
let me there.
Yep, looks good.
Try that. Okay.
Yeah, that looks even better.
All right. So yeah, thanks everyone for joining in. And Carl and I will go back and forth a little bit as we work our way through this. So most of our topic today is going to be around kind of what we've learned through extension work. There's this interest, like Carl said, I think over the last 10 to 12 years that we've been kind of playing with this project, that we've turned the corner on. Demand seems to be up. Locally grown hops, Wisconsin, let's just keep that as the local. Quality is definitely better. We've got some growers that have really embraced the quality part of this, and we're seeing that in the delivered product to the local brewers. So the way we got kind of started with this project is that demand for the local product. So they want local ingredients. Local craft brewers want that ingredient to be as close to their brewery as possible, especially within the state of Wisconsin. And so we've been able to, that's why we've had the interest. There was a demand there, and those of us in Extension are trying to meet that with some research. Also, there's that alternative crop side of it. I think some of our growers that we've seen over the past few years have either been farmers in the past or current farmers that are trying to add maybe a small crop to it, or they have someone in the family that's interested in trying something, or it's the way to expand into maybe a brewing industry or create their own beers. And so that diversification was part of the reason. And then maybe, is there an income supplement to what they're already doing? So a lot of our interest in this has come from the demand of the brewers wanting that local product, as well as maybe the farmer themselves are looking for another way to diversify or to provide some income. And so most of us that are in the beer industry realize that aroma and flavor is where that hop provides that for it. Those of you that maybe, I'm going to just go through some quick biology of hops, not spend a lot of time on it, since most of us are maybe aware of how they grow. But most of there's different families it's actually in the Cabaneaceae family, which is also connected to hemp. So those of us in Extension that have been working with hops and hemp, we've been in this cannabinoid area for quite a while now. So again, the one that commercial hops mainly grow are from the Humulus lupulus family. And so they're annual shoots that are on perennial rhizome, or perennial crowns, which is an extensive rhizome, but it sends up an annual shoot each year. So even though it's a perennial crown, we do have an annual shoot to manage. And so they are dioecious, where you typically have male and female flowers on separate plants, and therefore for production of hops, you typically are going to want to use the female flower to get those cones to develop. And most of those varieties are from cuttings or propagation that we tend to use. So again, this is just what it kind of looks like. The crown overwinters, you get this annual shoot that comes up, typically emerges in April. What's interesting is it does grow in a clockwise pattern. So when we show you a few slides of some trellising and that kind of thing, you wrap them clockwise, so they continue to climb up the string. And again, they can get quite tall by the end of June. So again, just a quick photo there of how the plant actually grows. What happens in the spring is this initial shoot can also, on a more established plant, that crown can be infected with some of the diseases, especially downy mildew, diseases like that. So typically we would try to cut this first flush of shoots. We would take those off to get rid of maybe some of that disease that's been harboring. And then you would do that about the first part of May, maybe, and then the plant would take off from there, and that's what you would manage. So we typically try to remove those early shoots just because they can be full of disease and virus.
Jerry, I could mention, if I could, just that out east, there is a little bit of an industry too with the hop shoots as a food source. They taste like a mild asparagus. There's a market in some of the gourmet restaurants out in the East Coast for them. Not a lot of people do it because it's a lot of work to clean them up and get them ready. But just to mention that there is a market there.
Sure. Yeah. Thanks, Carl. So around the first day of summer, that's usually when these lateral branches up towards the top will start to form. And that's where a lot of your production is. So the higher you can get that plant up into the trellis, there's typically more production the higher up into the plant. So then those female flowers, those cones, will start to develop after that summer solstice, or that first day of summer, so to speak. And that cone is where the lupulins are there. There's almost like a yellow powder, so to speak, and that's where the acids and oils are housed. And then you harvest that cone and use that as your flavoring for the beers. So again, they flower, form. Again, I guess I was showing the difference between the male and female flowers at that point. So once again, that's that lupulin that you peel those little leaflets back, and that's where you'd see the lupulin hiding underneath there. So that's kind of what we're trying to harvest, is that cone, and that's the product that we're going to be looking at for quality, as well as trying to keep that as disease-free as possible, since that cone is what we're after. So with that, I'm going to turn it over to Carl, and he's going to talk about, you want me to just advance slides for you, Carl?
Yeah, that'd be great. And just to mention, we don't want males out in the hop yard because we don't want seed formation. The seeds make the hops very, very bitter, and that transfers over to the beer and other uses. So they, in most of the country and in Germany, if you have, I can't remember the percent seeds, but they're rejected pretty quickly if there's any seeds available. But yeah, hops in Wisconsin, the return of commercial hops, as it says here, about 2010, several locations throughout the state put kind of the first hops back into the ground. There were a few grown in backyards and things like that, but that's when commercially that kind of started again after being absent since the late 1800s. Currently, and this is according to USA Hops, who does the survey every year, they got behind a little bit, but 297 acres is what was reported harvested in 2020. Some of the really small yards don't report, so this would be mainly the commercial yards. We may be a little bit less than that currently. We had a few growers that kind of threw up their hands, couldn't get up the quality that they wanted. But again, as Jerry mentioned earlier, it's coming back around again. Go ahead, Jerry. Kind of a cool slide if you look at the location of hop yards in the United States and Canada. The Pacific Northwest, Washington and Oregon, still produces about 85% of all the hops in the country. And you can see there's a few hop yards, but they're pretty good sized out there. The Yakima Valley is where the vast majority of that 85% are produced. You can see hop yards all over the Midwest and the eastern part of the United States, northeastern part, but they're very, very small in comparison. And really the commercial production is pretty low. Michigan probably being the leader after the Pacific Northwest, and they're still only at about 1,200 acres or so.
I just want to make a point. If anybody's asking why Goldie the gopher is up here in the upper left-hand corner, a few of these slides we borrowed from Josh Havel, who is a PhD student at University of Minnesota, and he just presented some of this information a couple of weeks ago at our hop conference. So Josh is allowed us to use these as long as we kept the gopher on there. So that's-
Yeah, I suppose we could have had boxing gloves on the little extension one and on the gopher
put-
one.
Bucky up there to fight the gopher. All right, go ahead, Carl.
This is just kind of real quickly, kind of the history of what we've done or some of the projects that we've done. We just held our 14th annual HOP conference a couple of weeks ago. We get quite a few national and international speakers and participants now that we've also offered it virtual. We had Canadian participants this year. We had Florida participants this year. So it brings an interesting mix since we added the virtual since 2020, right? Yeah, we had virtual. We planted a small hop yard in 2010 just to gain a little experience. We really didn't know if these things would grow or how they would grow. So we stuck some in the ground and they grew. Thus we said, hey, we can do this. A few of us did some study tours to Germany, Yakima Valley, and Vermont. Vermont was on our list because of their small hop yard that they have in northern Vermont, and also that one is managed organically. It's kind of cool to see how they've been doing with it. In 2012, we wrote a grant, Sarah Research and Education Grant, and were awarded that grant project. And with that grant, we studied both hop diseases with Amanda Givens and Michelle Marks, who was her PhD student. And then that grant also included malting barley, which we won't talk about today. Ruth Genger also participated with us in the Clean Hops program. I don't think, Jerry, correct me if I'm wrong, but I don't think that is available anymore. I think the funding ran out a couple of years ago. So, but it put clean hops, that would be virus-free hops, and into the hands of quite a few growers around the state. And really, I think as much as anything, promoted the whole idea of clean hops, not bringing in diseases that you have to manage immediately. And we'll go over a little bit of the diseases we're talking about and why in a few slides here. We've started doing fertilizer research in the state on hops. There's very, very little work that's been done anywhere other than the Yakima Valley on fertilizer. And even their fertility recommendations are 20, 25 years old. Really no work has been done out there since we conducted that. And we may be able to expand on that a little bit this year. I think we have a little bit of funding left that we will again do a small project depending on what we can find for funding. All of our research is done on private farms. We have no hop yards established on any of the university stations. We've had great cooperators with our hop growers in the state. Our quality has been a major issue, but current growers we got away from the hobby, and we're more into now the commercial serious growers for the most part in the state. And demand is, as Jerry mentioned before, is growing. Supply is short. And hop variety is always a big variable. There's quite a few hop breeders out in the world right now, both commercially and private, or public and private. Craft brewers are looking for a unique taste and aromas. That's what sells. And it's difficult for new growers. They'll ask, what should I plant? And it's really hard for us to give them an idea. And brewers are very reluctant to say, these are the hops I want. So it's kind of a tough entry into the market. Just a little bit on some of the hop disease research. Hop downy mildew is our most prominent disease in Wisconsin hops and across the country, including the Pacific Northwest. We had downy mildew showing up within 3 years after they were planted. Some of us probably bought rhizomes that had downy mildew already infected because there really wasn't a clean hop program when we first started this in 2010. Powdery mildew was first detected in Wisconsin in 2016. Not a huge problem. Our weather conditions tend not to be real conducive to powdery mildew. Fulma wilt is relatively new, 2018. It was discovered in eastern U.S. About that same time it was discovered in Wisconsin. A little harder to treat Fulma wilt, and it's probably the newest one we're dealing with right now. Newly funded, we have a newly funded USDA hop breeder, and they're going to concentrate a little more on disease management. Hopefully that's going to lead to better resistance, and the breeder is located out in Washington or Oregon. I don't remember which, but one or the other. Amanda Givens did a presentation for us that Jerry presented up at our last conference, and I really like these next two slides. One is timeline of disease management for young hop yards. That 0 to 5, the first 3 years in our established hop yards, really not a lot of disease if you buy clean stock. In years 3 to 5, downy mildew is going to get you, and sometimes powdery mildew by year 5 if it's available and the conditions are right. But downy will be there. It's very, very hard to. You're just going to get it. You plant hops, you're going to have downy mildew.
And the thing with downy mildew is it's systemic, correct, Carl?
Yes.
Where it'll harbor in the crown and can infect new plants or new shoots that
Right, and that's what Jerry mentioned. The new shoot, most everybody clips or mows off the first new shoots that come up. Some of us, as we move into northern parts of Wisconsin, or Jerry and I in west central Wisconsin, we'll usually get the benefit of a late frost to get rid of those. I have just about every year in my little hop yard, but there's very few systemic fungicides that will work on downy. The ones that tend to establish resistance pretty fast. Okay, then the next 5 to 10 years, there's quite a few other diseases that you're going to have issues with, and it requires a pretty rigorous scouting program and spray fungicide program. And we'll talk. We have a couple slides that'll talk a little bit about that, especially for downy. It's hard to get away from it. I've been to the hop yard in Vermont twice, and they're trying to control things organically yet, which means they use a lot of copper products, and they've developed copper toxicity in some of their plantings now because of too much copper in the soil. So there's not a lot of other organic fungicides out there, and some of the ones that were available aren't anymore because their toxicity levels were so high to the people applying. So it's the organic has been a tough world, and we can talk more about that later if anybody chooses to. Future needs, variety testing, both for quality, unique flavors, and disease resistance. The unique flavors is really, really getting to be huge. A lot of people will talk about the terroir in wine, getting way more popular to talk about that in beers now. And there's a group with one of our cooperatives that's doing a fair amount with sensory testing, just trying to identify some of those unique flavors from Wisconsin and from the region, really Wisconsin, Minnesota, and also use that as a guide for when to harvest. If you can train your nose to say, hey, I think this is the flavors, aromas that I want, boom, then they harvest. It may not be when the cone says this is time, it maybe this is the aroma that says the time. More of that will come in the future.
Yeah, the sensory testing we just did at the conference a couple of weeks ago. That was the first time I'd ever participated in something like that, and it was pretty unique the way that works with your standard aromas, whether it was mint or pine or whatever flavor you were looking for. You'd have kind of a baseline, and then you would say if it was more intense or not. And then you had to kind of put it in the right category with all these other aromas. And it was a very comprehensive, even though we went through it relatively quickly. I could see where you could spend a whole day just training yourself to go through that. So those people that are sensory trained or certified, that's quite a skill. It was pretty interesting.
And Jerry, unfortunately, for some of us, it works a lot better when you're a little younger. Your sensory does not get better with age, generally. So,
And it always smells better if it's in a beer, then you stick your nose in.
There you go.
We'll keep moving.
I'm just going to go real quick through this. This is a little information. Jerry mentioned Joshua Havel. He might be defending his PhD next week, so he's pretty much done with this program. But he's done a lot of work with wild hops and genetic characterization of hop germplasm throughout the country. Actually, we've had lots of feral and wild hops from Wisconsin that were entered into his testing program. Go ahead, Jerry. And I'm only going to mention just a couple things here.
Oh, sorry.
Oh, wow. You really want me to go fast, Jerry.
Got a
Okay.
little
Let's
punchy.
speed up a
All
little
right,
here.
hang on.
Okay.
All right, there. I wanted to
There
make
we
sure
go.
there wasn't a highlight.
We're mainly just looking at the common hop. Joshua has categorized where different germplasm has come from. The common hop from North America is the ones that he concentrated on for this program. Go ahead, Jerry. And hop production is limited by powdery mildew. We mentioned that briefly. It causes the powdery mildew on the left side, and then really cone degradation on the right side. Okay. And this is, we don't have a lot of powdery mildew, but throughout the country, you can click and bring up the next ones too, Jerry. About 15% of all the hops are lost from powdery mildew. The alpha acids are really greatly reduced by powdery mildew, and about 80% of all the U.S. acreage is susceptible to powdery mildew. You could say the same thing basically about downy mildew as well for Wisconsin. 7 to 10 fungicide applications per year are common, and cultural management. There are some cultural things you can do to avoid maybe 2 or 3 of those fungicide applications, but they're very labor intensive. And as most people know, labor has really been difficult since 2019.
And Carl, it's pretty much on a 10-day, 7 to
about every 7 to 10 days, depending on the weather patterns.
Yeah.
For fungicide applications.
That one kind of came up towards the bottom there, Carl. There's another bullet on.
The genetic risk. They're working hard, but you're looking at a 10-year cycle to really see if the hops you're producing and breeding are going to be effective and have the other qualities that you want besides the genetic resistance. Go ahead, Jerry. Next slide. They have identified the QTLs associated with powdery mildew. This is part of Josh's work. The thing right now in the brewing industry, the same holds true for malt. The industry will not allow for GMOs. So it's a little harder to once you even have the QTLs identified, to try to get those into the varieties that you want. The CRISPR technology, which I don't really know, maybe Jerry can explain it, or maybe we'll just not try to do that today, may be allowed in the hop and barley industry, but as of this point, it's not.
Yeah, the CRISPR technology is basically manipulating that, turning that gene on or off. I think is what they're basically. It's not the genetically modification, like putting in another species like you do with a BT corn variety. It's actually manipulating the genes within the species itself.
Okay, go ahead, Jerry.
Oh, you got a lot of little arrows here.
Yeah, there we go. Let's pull all of these up and then we'll talk about it. Well, about those two. I'm just going to mention 2 of the conclusions that are probably the most effective or important for Wisconsin right now. The resources for powdery mildew resistance. There have been some of the feral and wild hops that have been identified as having pretty broad spectrum resistance to both powdery and downy mildew, which is super important for the industry. And then the last conclusion, that there's way more potential for this hop genetic resources for understanding the traits that those could bring into the program. Not a lot of money has been, Josh has been the only one that's really working on the the wild hop genetic resources, and he has funding for a couple more years as a postdoc because he'd like to stay in Minnesota for quite a few different reasons. But it's still really hard with a one-person shop with maybe a couple students working with him to make many strides in the program. He does have a couple, or Minnesota does have a few different varieties that are not commercially released yet, but we have some growers in Wisconsin that are growing them out, 5 to 10 plants, just to see how they react agronomically. And then there's some brewers that will participate with them to do some microbrews to see how the quality is with them. I bring one thing I just want to mention from a hop program that we had in 2019. We had a researcher from the whole research station in Germany, Wallensack, Germany, present, and he talked about how much pressure they are in Germany to deal with the number of fungicides they're applying. So we may be able to draw more of the of some of their genetics into the United States as time goes along. They're probably 10 to 20 years ahead of us in breeding for disease resistance in Germany, just because of the pressure for getting away from that many fungicide applications. So time will tell. And I know Josh is also working with Florian is the researcher's name that we had talked. And Dave also are in contact. So hopefully that will move ahead a little faster than it has up to this point. Okay, Jerry.
Yeah. Is there any questions for Carl or I regarding this section of what we're going to what we've discussed? I can't see the chat box, so
Nothing in the chat yet, Jerry. Okay. I got it up. And if you want to go to the credit slide, I'll just
yeah,
a couple things.
Sorry, we had one more.
The acknowledgements, it's pretty broad, the cooperation that Josh has had for his research. Hopsteiner is one of the larger hop organizations out in Yakima Valley, and Washington State has been pretty involved with him as well as among the other local sponsors. So I just wanted to point that out in Joshua's credits.
Okay, thanks, Carl.
Just a quick question. I'm curious, like, what the price point is for Midwest grown hops and how that compares to hops grown out west.
That is another project that we're still working on. We have 1 year of data, and it's something that we'll be sending out real soon to hop growers, because that is a huge question. And right now, we don't have enough data even really to respond confidently. So it costs us a lot more. I'll give you one example. To pellet hops in the Yakima Valley, it costs them about 20 cents a pound or so to pellet. In Wisconsin, that's $1 or more to get them pelleted, just to do the pelleting process. So the volume disadvantage or advantage, however you want to look at it, is pretty big right now.
Yeah, but brewers are still willing to pay more for the hops.
Yep.
Okay.
Some brewers are, not all brewers. It's one thing that's also a little frustrating for those of us that have worked in this project for a number of years is that we're getting there right now, but up until this point, we've never had the close connection between brewers and hop growers as they've had in other states, specifically out east. And part of that, and maybe if Robin Shepard could even respond to this a little bit, as he's pretty familiar with things in the beer industry, but I categorize it as Wisconsin has a lot of blue collar beer drinkers yet. And they're willing to pay maybe $3, maybe $4 a pint for a craft beer. But you get out in the East Coast, they have no problem with paying $8 to $10 a pint, or even Minneapolis, St. Paul, or maybe Madison is maybe a little closer to that, but throughout the state, that's not really true. And so price of the hops has been maybe a little bit more important for craft brewers here.
Yeah, the other thing too that drives that, Stefan, is the variety. So if there's a specific variety that's in demand, that could be, or it has a specific aroma, or it does something specific for that beer that brewery wants to develop or needs that, currently needs, that'll drive that price as well. So it's always demand. Like anything else, it's a demand type of thing, but the quality's got to be.
I do wonder, Carl, about the whole push for, hyper local and what that will mean in terms of willingness to pay, what that's going to mean for, locally generated hops, locally grown hops, as well as maybe even on the barley side too, because, I think this whole interest, like you said earlier, the terroir of wine, the idea of the beer being tied to the local community in a variety of ways means the consumer is willing to pay more. And it may mean that, the hop prices, local designer hops will, some small brewers may not blink an eye at that if they can market it as a local product.
And great point in both Jerry and Robin in those remarks. And one of our larger brewers, probably more well-known craft brewers, is New Glarus Brewing, and they're really looking and working with the hop associations now to get those local hops with a unique flavor. And that's something relatively new that they're willing to do. There are quite a few of the other brewers that around the state, if you look around a little bit, that are paying premiums for that unique flavor. And I think the more our hop growers know and experience the whole sensory effect and train themselves, I think the more they'll move forward quicker with that.
You also reminded me, Carl, of the story of Pearl Street and the Linalool IPA.
Yep.
It was all based on sort of an heirloom hop that was discovered on Paul Stram's farm here in Dane County. And Pearl Street virtually for several years locked up all of that hop that they could get and named the beer after the compound. And so and I think that was sort of reflecting of that locally grown movement.
Mm-hmm. Good point. Thanks, Robin.
All right. Any other questions on that topic? Otherwise, we'll keep moving through our segment here. I'd like to kind of just put this section up, why I got involved in it. Well, I'm based here in Chippewa County. And so there's the Leinenkugel Brewery, there's the Leine Lodge, and there's my office, all within about a block of each other. So I have to kind of work on hops if I'm that close to the brewery. What I'm going to just present is some of the soil fertility research that we've done. Carl kind of alluded to it earlier that there wasn't a lot of data or local data, even from Wisconsin, that related to especially nitrogen management. And so most of our recommendations were based off of the Pacific Northwest, and we worked pretty closely with Chris Baxter. He's our soil scientist with UW-Platteville, but he's an extension soil scientist, as well as Carrie Lebowski, who is a former UW-Madison soil scientist and now moved out to Penn State with the ARS project out there. But we've done a pretty intensive soil fertility research project, primarily around nitrogen, starting to work into potassium at this point. But in 2016, we kind of just took some plants, harvested a few, and tried to get a baseline of what is the demand of the crop as far as what's some soil testing, and then just took some leaf and tissue samples, measured yield, and tried to get some of that data so we could move forward. In 2017 and 2018, we did some more data collection on a more extensive level, where we started to look at about 3 locations across the state, looking at getting a protocol, so to speak, kind of fine-tuned, looking at biomass, cone yield. And then we also incorporated some of the petiole nitrate testing, which was relatively new for the hop industry, looking at it from, is it accurate, along with tissue testing. So then we had another 2-year study to look at the optimum N rates here in Wisconsin. So that included much more intensive work, and it really got a call out Ken Schrader in Portage County Extension, who led the charge in that area, in the Rosholt area with the grower, as well as George Kep. He was down in Columbia County, worked with the grower there. And then Bill Halfman was in the Monroe County, and he had a location as well. So Ken and Bill and and George were the kind of the on the ground instigators, or the people that made this project go. But we included 5 rates. Again, a lot of testing in terms of tissue and sap, and we evaluated the nitrogen rate based on yield and cone biomass and cone yield. And then, did the usual sampling to try to estimate if we're in a good spot, or what that recommendation for nitrogen could be. Carl's got a bunch of those updates on his website, but then just recently here in the last couple of months, we had this research was published in the Journal of the National Association of County Agricultural Agents. So the work that we've done on this has been published and is available for you to thumb through if you're interested. So just going to kind of thumb through. Is this? Can you guys see all of that, or is my thing in the road there? You're okay? All right. I'm going to move this screen a little bit. All right. So this was the biomass, the nitrogen effects on the biomass. So we looked at 7 different varieties, and even though we couldn't do the same varieties the 2 years at the 3 locations, we tried to pick those that were similar in growth pattern, or at least similar in the demand for oil and use and that kind of thing. So the way this chart is, we got 2019 and 2020, and then we have the 5 rates that we use. So we had a check, which would be the 0, where we didn't apply any nitrogen, all the way up to 200. And the way the project was set out was they were replicated 4 times across the yard within that specific variety. And so we treated 5 plants and harvested the middle 3 to try to avoid the border effect. So that was the way that the project was laid out. In general, I think you can see in most cases it trended upward, where some nitrogen was needed as we moved through both years, that there was a response by most of the varieties. That is, as you increased in nitrogen, there was an increase in biomass. Same with cone yield. You could see that the 0 rates definitely had some of the lower cone yields, and then as we increased nitrogen, it continued to rise. So that was kind of the year by year, variety by variety, and with a lot of this, the variety is very reacts much differently from variety to variety on nitrogen application. So when we look at the biomass across all the locations, you can start to see, okay, this is kind of where, based on having those 5 rates, you could see where things started to level off. And in some of our earlier surveys of growers, they were usually around that 200 pounds per acre. That's kind of where the rate was coming out of the Pacific Northwest. And so with Chris Baxter's work and his guidance, we settled on these 4 application rates with the 0 strip. So on biomass, you could see that was climbing and then kind of leveled off around that 150 pounds per acre. And then we looked at, okay, what does that mean for cone? And so the cone yield was pretty much the same as well. So in most cases, we got to about that 150 on the cone yield, and then it started to taper off. So where biomass started to kind of level off, we saw a little dip in cone yield that, that extra 50 pounds really wasn't producing any more biomass, and it really wasn't producing any more cones. And so from a statistical standpoint, we see that these 2 are statistically similar, but there was a significant difference, the more or the less nitrogen that was applied. So part of it too was looking at the petiole and sap tissue analysis and those concentrations using 2 different methods. So with this, there was the sap taken right out of the petioles. A certain number was collected, sap was squeezed out, tested, and then tissue samples from those same plants were sent to a lab. And so there's a pretty good correlation here between the cone yield or the tissue petiole sap concentration and the tissue concentration. I don't know why my labels didn't show up there. So you can see that they correlate pretty well. I think it was about a 0.7 correlation on this. So that part we felt was pretty strong as far as Chris was comfortable that the petiole sap testing, that is something the grower could do on site on their own and tell if they've got enough nitrogen within the plant. The other thing we looked at was the hop storage index, and that's something that is basically the measure of how long will that hop store over a certain period of time without losing the alpha and beta acids, and it kind of has to do with handling and the long-term storage of those hops. And so you could see that the hop storage index was a little bit higher with those lower rates than at the higher rates. And basically that meant the higher rates, that storage index, I think, Carl, if I remember right, the lower the number, the better it was, that it stored better, or have I got it inverted?
No, we want a higher
Higher
hop storage index. So excess nitrogen actually decreases it. All the rest of them were statistically different.
Okay. So the excess nitrogen is what was bringing that down, and probably had more to do with the oils being just the handling. There was more to handle, and it was degraded more that way. So a few things we're going to be working on here in 20. So that grant ran out. What else could we look at? We had a few dollars left in some grants. So we looked at potassium as kind of a baseline, similar to what we did with nitrogen, and looked at one location in Rosholt, again, where Ken Schrader was located. So he was able to put that on, the potassium on. So we looked at potash at 150 and 130 pounds of nitrogen, of potassium. So, we know that it's pretty important for photosynthesis, some of that winterization of the hop. So we know that potassium is a major nutrient. And so it also helps to activate some of these enzymes that are for that energy utilization. And there's also a major interaction, being a cation, that it's going to move some of these other positively charged ions around. So just in this first 1 year data, we can't draw too many conclusions, but it's kind of leading us to where we're headed in the future, possibly, is that the potassium really didn't have a major difference in so I guess we should back up. This is the potassium rate down here, where we've got the 0, 50, and 100, and this was just the difference in the varieties that were there. And so based on, there was a big difference in, this was the baseline between the biomass, or the bines and the cones between the varieties. So very similar reaction between these two varieties. But then when we looked at it from the application of the potassium, you could see still around that 100 pounds started to be kind of that higher rate. Going any higher wasn't making any difference. And it actually dropped yield. And Chris felt that had something to do with knocking off some of those ions of calcium, and therefore maybe displacing that ion and dropping yield from that standpoint. So from a cone quality standpoint, the variety basically was what was affecting that total oil and all of the components of it, which is what we would expect. And so looking at it from that standpoint, the varieties, basically it was too much noise in the data to really, it was probably more driven by the variety reacting to that potassium than any of the, it was more variety driven than by the actual application. So again, the treatment had significant effect on hop storage. Again, you could see it dropping at those higher rates again as we move through the different varieties with the pounds of potash that was applied. And so there again, those higher rates tend to have a significant difference on the storage index.
Hey, Jerry.
Yeah.
Jerry, and you were right. You want a lower number. You had it correct. I was wrong. I just looked it up.
Yeah, I thought it was inverted, but that's okay.
Yep, you're correct.
So so here again, you're seeing a lower number with maybe that lower rate of potash. Again, wasn't significant in Magnum, but more in the Willamette. So that part of it, just seeing that there's probably just a variety difference to it. It's not huge here within the hop storage index. But again, there is something going on with the potassium application at that lower rate, specifically with Willamette. So that's kind of where we think we're going to head with this. We kind of got nitrogen figured out that 150 pounds seems to be more the sweet spot than 200 is. And then what does that mean for potassium? And so some of these observations, again, it's going to play an important role. We kind of know that information. Again, soil, that sap testing, if farmers can, growers can use that petiole testing on site. They can do some management more reactively than wait for that soil and tissue test to come back. So sap testing, we're hoping, has that potential to kind of be used similar to what nitrogen is. On average, usually if you're in that optimum range of that 120 pounds per acre, something like that, that's usually going to optimize your cone yield. But it can, if you go over that, it could displace some of that calcium ion in there, and that could be driving down some of that yield as well. So again, could slightly decrease the cone yield and then have a negative effect on that hop storage index. But again, for 1 year data, just exploratory, we're hoping that maybe we can move into the future and do a little bit more with it. I'll wrap up here with hop, some of our extension impact on this. We've used this data during some educational programming, again, mainly with our, we usually have some annual field days and then the hop conference, but just doing some survey of producers that attend these on a regular basis. We got 12 responses out of the growers at our conference, and they've indicated they've reduced their application rate by 50 pounds, which we think is a pretty significant savings for hop growers. It's not huge, but when we look at it from an environmental standpoint and also a farm profitability, if we can lower any input and still get the same yield, they're pretty happy with it. So that's kind of where, that's kind of a feather in our hat that says we've made a little impact in this industry by growers taking this research, on-farm research data from these 3 sites and saying, yeah, 200 pounds per acre is probably too much. Let's back off, and they seem to be happy with it. Most of our conferences attended by 30 or more participants on average. I think when we first started, we were probably in the 50-person range pretty easily, and we've kind of settled into that 30 participant range over the last several years. And then we did have the journal article published just this last year. So we've had some pretty good impact from the extension side of things. So with that, I'm going to wrap it up. Carl's on if you've got any questions, and I'm going to quit sharing my screen here so we can have a discussion if we need to. Oh, Bill's on. Didn't see Bill pop in.
Cool. Thanks, Jerry and Carl. That was great. I'm going to stop recording so we can just kind of have an informal